Hey, hello Bio, this is Mr. B, and we're starting the second semester off with Chapter 19, and it's entitled Bacteria and Viruses. So I would know this for the quiz that the title of Chapter 19 is Bacteria and Viruses, and I know we're going a little bit out of order, but this is kind of a nice chapter to start off with because it's interesting, and we've kind of hit on what bacteria and viruses are, but we haven't really gotten to the specifics, so I feel like this would be a good way to start off the second semester. And so to start, we're just going to go over what bacteria are. So that's what we're going to start with, as our focus is going to be on bacteria. And to start, we'll give us some background information, some review, is that the smallest and most common microorganisms, again microorganisms meaning just small organisms, are prokaryotes. So we've talked about eukaryotes and prokaryotes several times throughout the course of this year. And we just need to review that prokaryotes are unicellular, meaning one-cell organisms that lack a nucleus. So bacteria are going to be just one cell, and they're not going to have a nucleus, so they don't have that centralized control area that that are eukaryotic cells, which are humans, plants, animals, fungi, things like that. So these are just unicellular organisms that are going to lack a nucleus. That's what we need to know about prokaryotes. Another thing we need to know about prokaryotes is how they were once classified or grouped together. And way back in the day when we first discovered prokaryotes, they were placed into the kingdom Monera. So that just means we haven't really gone over the levels of classification yet, but to know what this means is that way back when, all bacteria used to just be placed in one group. So at one point in time, all bacteria were in a group known as Monera. However, today, biologists have divided them into two different kingdoms, or two different groups, known as the Eubacteria and the Archaea bacteria. So those are our two groups, Eubacteria and Archaea bacteria. And so the focus of the rest of this video is going to be on the difference between those two groups, and as this slide says, how do the two groups of prokaryotes differ? So first off, being our Eubacteria, we're going to take a look at the characteristics of these. Eubacteria have a cell wall that protects the cell and determines its shape. So if you remember, we talked about there are some plant cells that have a cell wall. So we already know what the function of a cell wall is. It's no different in the bacteria than it is in the plants, really. It's provide protection and it'll determine the shape. And a new word is that the cell wall of Eubacteria contains peptidoglycan. Now this is a long word, it's a mouthful, but all we're going to need to really be able to do with this word is when you see this and seeing that, if I wrote down on the quiz, let's say, which type of prokaryotes have a cell wall that contains peptidoglycan, you would just need to know that it's Eubacteria. We don't need to know what the makeup of this molecule is. We just need to know that it is found inside of Eubacteria cell walls. So our next thing about Eubacteria is they have a cell membrane that surrounds the cytoplasm. This isn't anything new. I think every cell we've talked to up to this point has had a cell membrane that surrounds cytoplasm. However, this next one is going to be something that's a little bit different, is that some Eubacteria will have a second membrane that provides added protection. So we've talked about plant cells, and they have a cell membrane and they have a cell wall. We've talked about animal cells, let's say animal cells and human cells that just have a cell membrane. So now these prokaryotes, these Eubacteria, have two cell membranes and a cell wall. Now that's not all of them, that is just some of them. And that is kind of a big defining characteristic of this group. And this next slide is just a picture that you guys already drew in class of what our typical prokaryote will look like. So this is what E. coli is an example that they give, and it just gives all of the different structures and words that we will learn about throughout the course of this chapter. And so then our next slide, we're still going to look at where Eubacteria live. So these include organisms that live in a variety of environments, and these can include in fresh and salt water. So pretty common, we know that bacteria live there. They live on land, normal, bacteria live everywhere, and in the human body. So I know I've mentioned before that we have billions, if not trillions, of bacteria living in and on us at any time. And all the bacteria that we find living on us usually are going to fall into the Eubacteria. So that's the Eubacteria. So we've given kind of their characteristics and where they live and what they look like. So our next group is called Archaea bacteria, and this is a very interesting group, and we're going to kind of look at what sets them apart from Eubacteria, why they're separated into two groups. So first off, they do not contain peptidoglycan, so that's a big characteristic there. So the other, our eubacteria, did contain peptidoglycan. Archaea bacteria do not. That's one difference. And they have different membrane lipids. So again, that just means lipids being fats. They have different fats laying in their cell membrane. And also, in addition, the DNA sequences of the archaea bacteria are more like those of eukaryotes than those of eubacteria. So when we look at their DNA, they're very different. If we we don't have the technology to do that, but scientists today have the technology to take a look at their DNA and say, wow, these really aren't anything alike. So that's kind of been their big distinguishing characteristics. And the last slide here for this video, very interesting, is that many archaea bacteria live in extreme environments. So these extreme environments include things like not just on the human body, or not on land, or not on water, things we wouldn't think of life being able to be sustained. So there's examples they give us are methanogens will live in oxygen-free environments such as thick mud and animal digestive tracts. So there are living things that live in areas where no oxygen is present. And other archaea bacteria live in salty environments or in hot spring where water temperatures approach the boiling point. Again, these are things that they are able to sustain life in areas where we wouldn't ever think it would be possible. And we're going to take a more in-depth look at those in class. So that's it for this video. Let me know if you have any questions.